Good morning, everyone. Today, the idea was to talk a little bit about the last things that we've done in terms of infectious coryza in the lab. There's a couple of things related with the persistence of the bacteria, and also some research that we've done on inactivated vaccines that are available in the market in order to see, the load of antigen that they have, and try to associate that with effectiveness in the field. I think there's a comment in the chat. Oh, okay, sorry, it was an answer to a question. So this is the agenda for today. So generalities about coryza persistence, first of all in the poultry environment, and then in treated and untreated water, genotyping, and vaccine testing. So infectious coryza is caused by this bacteria that has a very terrible name, which is Avibacterium paragallinarum. It's a bacteria that it's really difficult to grow in the lab, but according to the textbook, it's really easy to kill in the environment. Lately, we've seen that it's not such a case, and maybe that's because this bacteria changes. And actually, after doing some research on the genome of the bacteria, we've seen that it changes quite a bit, more than what we thought. Anyhow, we'll talk about that a little bit later. In the US, the places that are, or the states that are enzootic to this disease are California, Florida, and Texas. It's usually associated with temperate climates and also with lack of biosecurity. As it says here, biosecurity breaks are involved in the introduction of the bacteria and the carrier stage. So meaning the hens that get affected or infected and then recover are related with the persistence. So these hens that actually suffer the disease and then recover will be shedding the bacteria throughout their whole life, and that's important because because of the reality of layer farms in the US, in which we have multiple ages. So if your farm gets positive, most likely will be positive forever, and that's why here in the West Coast we use vaccines. It causes upper respiratory signs such as nasal exudate, swollen sinuses, and depression. It also can affect feed consumption. In some cases, approximately 12 pounds per 100 birds per day, which is a lot considering that laying hens in production eat around 110, 120 grams per bird per day. And 12 pounds is like 70 grams, so it's a lot. Up to 70% reduced egg production. Usually, it doesn't affect that much. We saw that some years ago, usually would be 10, 20%. And when complicated with E. coli, IBV, ILT, or MG, they can cause septicemia and mortality. So this chart I always show because I think it's important to realize that diseases in general in chickens are not caused by a single agent. They are multifactorial. When coryza is complicated by other infections such as E. coli, Pasteurella, Mycoplasma MG/MS, pox, IBV, and ILT, you usually have a chronic course of coryza in which you have the clinical outcomes will depend on the complicating agent. Whenever this coryza is uncomplicated, meaning that only the Avibacterium is causing the disease, you have an acute course with 1 to 3 days of incubation, 7 to 14 days of signs persistence, and sometimes the signs are not very noticeable. And we see that sometimes in the Midwest, in places that are cold, and usually the comment from producers or veterinarians is that they have positive PCRs for coryza, but they haven't seen any clinical signs from the disease. The important thing and the bottom line is that the outcome of these infections, either complicated or uncomplicated, are carrier hens. So these hens, again, that are PCR positive, that are healthy, but they're shedding constantly the disease because they got it and they recover, right? So in terms of transmission, we have direct contact with infected chickens, chronic healthy carriers, so these persistently infected birds, Biosecurity flaws, which is related with the introduction. We're not sure, but we think it can be airborne transmitted, associated with humid weather or water. Maybe it stays in the water for a longer time. And anecdotally, we've seen more cases in winter and spring when the weather is foggy and when orchards are spread with poultry manure. And also, especially in broilers, we see the disease approximately 10 days after windy days. So this is an experiment that we did in 2019, I think, but it's to illustrate the way in which this bacteria can persist in poultry environment. So what we did was to get some broilers that were previously infected with coryza. We stuck them in some isolators, infecting the whole isolator, the environment. We also brought bedding material. And after keeping them for some days, we eliminated all those broilers and we brought naive birds in order to get exposed. Well, long story short, we saw that coryza was detected in the cedar birds, so the birds that we brought in order to contaminate the environment, in addition to IBV and Gallibacterium. So again, informing that this disease is usually not a single agent disease, it's a complex disease. We did not see coryza PCR positive exposed birds, meaning that the bacteria wasn't able to persist in this environment. And finally, these exposed chickens showed mild clinical signs such as swollen heads, mucus exudate without the presence of the avibacterium. So what this means is that other than coryza or this avibacterium, there's other pathogens that are also part of this clinical condition, which are swollen heads and all exudate coming out from the eyes, etc. So meaning that in most of the cases, this disease is complex. Now, after knowing that, we started doing some experiments looking at the persistence of this bacteria in water. And the idea was to test the persistence of the bacteria in 2 different temperatures. So for these experiments, we hypothesized that the water at low temperatures increased the Avibacterium paragallinarum persistence. In addition, chlorine at 3 and 10 ppm is able to kill coryza in spike water. So that was our hypothesis. And the objectives of the experiment were to determine if the variability of coryza, if the viability of coryza in spike water at 5 and 20 Celsius at different time points, and also determine the effectiveness of chlorine treatment in the water at 3 and 10 ppm in the inactivation of coryza at 5 and 10 Celsius. 5 and 20 Celsius, sorry. So this is the experimental design. We had 2 boxes with 3 beakers each. 2 of those were positive and spiked with 100 million bacterial colonies or bacterial cells. And then we had a negative control beaker, non-infected. One group was at 20 Celsius, the other group was at 5 Celsius. And the samples were collected at 0, 30, 1, 2, 4, 8, 16, 24, and 64 hours in trials 1 and 2. We did cultures because we wanted to know if the bacteria was alive or not. And those cultures were performed with chocolate agar and incubated at 37 Celsius with 5% CO2. And and as I said before, each positive beaker was spiked with 100 million cells of coryza. And what we see here are these 2 graphs. On the y-axis, we see the colony forming units per plate. So basically the amount of bacteria that we can see in the plate. In the x-axis, we see the hours, the same for trial 2. So in trial 1 at 5 Celsius, which would be the blue line, we can see that the bacteria was able to persist throughout the whole experiment. Actually, the last time we checked them was 24 hours and still persisted. On the contrary, we have at 20 Celsius, the bacteria, the viable bacteria got reduced and disappeared. We didn't find more viable bacteria around 24 hours. When we repeated this, we saw that again, at 5 Celsius, the bacteria was able to persist until 64 hours, which was the last time point we checked. And the bacteria at 20 Celsius was able to persist until less than 40 hours. So at 40 hours were dead. that I think it's very informative in terms of biosecurity and in terms of the way in which we clean and disinfect, because these bacteria from textbook information saying that it's very labile, so where it's easy to kill, it's not that easy to kill if it persists until 24 in one trial and until 40 hours in the other trial. And actually, at lower temperatures, you're not able to kill it, right? So it persists until 64 hours. So we repeated this for a third trial, checking again the viability at 20 and 5 Celsius, but in this case, we added some groups that had 3 and 10 ppm of total chlorine, right? Chlorine is one of the things that we use in order to potabilize water or to disinfect water, consumption water. So we wanted to know if it was effective in order to kill the bacteria. So in this case, the samples were collected until 108 hours for bacterial culture. And the same thing as before, each beaker was spiked with 100 million bacterial cells. So this is the experimental design. We have one positive beaker with 3 ppm chlorine total. Then we had another beaker with 10 ppm chlorine total, spiked with the bacteria, one positive control also spiked with the bacteria, and one negative control that wasn't spiked. That was at 20 Celsius. The same was repeated at 5 Celsius. The water pH was 7, approximately 7. And as an information, and you guys know this better than me, it is recommended to chlorinate water in poultry houses to obtain 2 to 3 ppm of free chlorine. That's why we used 3 and 10 total chlorine, thinking that 10 will end up being 3 whenever you measure it in the field. So this graph is showing the results. So we have on the y-axis the colonies or the bacterial cells, live bacteria that we found, and then the hours on the x-axis. So if you take a look at the chlorine-treated groups, those were able to kill the bacteria pretty quick. Actually, the highest persistence was found at 20 Celsius with 10 ppm, and it was less than 12 hours. So there's a good action of chlorine against coryza. But then when we take a look at coryza without treatment, without chlorine treatment, at 20 Celsius, the bacteria was able to survive less than 36 hours. And when we take a look at 5 Celsius, we saw that this bacteria was able to survive until 108 hours. And actually, at this point, we found 3 colonies. So again, that's reinforcing the idea that if at certain conditions, the bacteria is able to persist for long periods of time. I mean, 108 hours is a lot of hours. Now, moving towards molecular characterization. So we know that serotyping is not available here in the US, and it's very difficult to find laboratories that do this in the world. Because of that, we have focused on studying genotyping here in our lab, trying to associate the genotype with the serotype. So we know there's 9 different serotypes divided in A, B, and C. So we want to have a system in which we can do some molecular biology, and with that information tell you if you're dealing with an A, B, or C strain. For that, we've been using this gene, HMTp210, which is the hemagglutination gene. When you do serotyping, you do hemagglutinations in the lab. And we think that this gene is the adequate in order to look at their sequence, because that's the gene that performs the agglutination. So one of the things we've done is to, most of the literature that it's available on this focuses on one segment that due to our investigations, we think it's the wrong segment. So we've been looking at longer pieces of this gene in order to do our associations. But then the other problem is that there's not many sequences in the databases. So currently, so we started contacting different labs in the world in order to get more sequences that are representative from the different serotypes. And now we're collaborating with laboratories in Indonesia, Argentina, Colombia, Netherlands, and the US. So the idea would be to have all that data, which we're working on. Actually, we're pretty close to have something definitive, and be able to type them. Now, why the typing is important? Because it seems that there's a lot of lab work and genetics, but it doesn't really apply to anything that we can do in the field. It's important because of vaccine guidance. So maybe the vaccination can change if you have a different type affecting your flock. The variability of the bacteria, it's really important to surveil the variability of the bacteria in terms of knowing if there's something that we're doing that it's actually making this bacteria change more quickly. And also the epidemiology. So it's important to know from where it's coming, right? In order to take action in terms of biosecurity to prevent this bacteria to come into our flocks. So as a matter of information, we only thought that the US had C strains, but by now, because of this collaboration and because of the work that we have done, we know that there's also a high prevalence of A strains. Here in the West Coast is usually C. We haven't seen anything different, but in the East Coast and some parts of the Midwest, we've seen A strains. So it's important to know that in order to prevent it appropriately. So again, this is a little bit more of the same. We're focusing on a segment that it's usually it's similar. It's close to 2/3 of the gene, and again, we're gathering all of this information. This is part of what I was telling you before. So these are all the strains that we have been able to gather, and they're color coded. So red would be C1, this chunk here. And we don't have any strains from the US that are C1. There's a bunch from Taiwan, France, Colombia, Japan. That would be H18. That's the type strain for C1. Blue is C2. This is where all the strains from California are located, and some strains from Pennsylvania, some others from Virginia, and our California strains. Then we have C3, which is yellow, in which we need to work on getting more sequences because the only sequence that we got is from South Africa. And then we have green, which is B. Orange, it's A1, in which we do have some strains from the US, specifically from Florida. No, these are from Nebraska, I think. And then A2 in purple, which are sequences from Florida. So again, I mean, you see that with this, we're able to have more information in terms of which strains are affecting different locations, which is important for control. The other thing that I have to say also is that we've seen that this bacteria mutates really fast. And even though our strains are still C2, we have seen some recombinations and some changes on some of the strains that we have obtained. I don't think those are affecting the effectiveness of the vaccines at this point, but it's something to take into consideration for the future. And the last topic that I would like to touch is this vaccine testing that we do here. So basically what we do, we grab killed vaccines and we run PCRs in order to know the load of antigen that those have. That doesn't mean that it's because by standard, most of the companies add more than 10 to the 8 bacteria into their vaccines. But we've seen that when we check them, there's something in the process that disintegrates most of these bacteria, meaning that there's less antigenic load and probably less immune response or a less effective immune response. So from all the vaccines that we have tested that are here in this slide, we have that they go between 10 to the 3 to 10 to the 8. So other than we really want to know why that's happening. And in order to understand that, we have done comparisons between bacteria that have been that are live and bacteria that are formalin inactivated, which is the way in which you put them in inactivated or killed vaccines. And as you see here, this is a picture, electron micrograph picture. You see that the chromatin on these bacteria is a little bit more dense. There's some membranes here, but there's not real difference between a control culture, live culture of this bacteria, and formalin inactivated bacteria. So probably it's not related with the formalin inactivation. So what we did in order to continue our investigation was to centrifuge these bacteria, and in order to centrifuge these vaccines in order to get 3 layers. One of them is the oil layer, which is in the top. The second is an aqueous layer, which is in the middle, and then a solid pellet, which usually contains most of the bacteria. After doing that, we selected the aqueous layer and the solid pellet in order to do electron microscopy. And this chart or table is showing the presence of intact bacteria, broken bacteria, and membranes from 5 different vaccines that we tested. So overall, we see that most of the bacteria are in the pellet from vaccine 1, 2, 3, 4, and 5. But some of them, for example, vaccine 3 doesn't have much antigen in the pellet. That might be related with what we're trying to figure out here, which is some vaccines probably use an adjuvant or a process that is actually disintegrating the bacteria at the point in which you cannot even recognize. I would like to show you some of these pictures. So this is for vaccine 1, in which we see a lot of bacteria. Vaccine 2 is pretty similar to vaccine 1. Then we have vaccine 3, in which there's areas in which we don't see bacteria. So definitely there's something there. Vaccine 4, we see a lot of membranes and pieces of bacteria. Vaccine 5, similar to vaccine 4. And then in vaccine 6, we did not see any bacteria. So again, this is not conclusive, and it's just to show you that we're doing, we're trying to understand a little bit more these vaccines. But the point that I want to make is that not every vaccine is the same, and in addition to that, you know, it's not only the vaccine; it's also the way in which you apply it. So I would encourage you guys to be present whenever these vaccines are getting applied. Check takes, make sure that the vaccines are correctly applied, because that's a key point whenever we wanna vaccinate and we wanna immunize. There's a difference between vaccination and immunization. Immunization will be generating immunity in order to protect against the pathogen that you're using in the vaccine. That's the goal. The goal is not vaccination. Sometimes vaccination is not well done. So in conclusion, infectious coryza does not persist in bedding, but it does in water. Persistent carrier state of recovered hens allow the bacteria to persist in multiple age flocks. Additional research needs to be performed on typing methods for these bacteria. We're fine-tuning our typing method. Coryza clinical signs mask other challenges that require attention: IBV, ILT, MG. So don't only focus on coryza. Just look at the flock in context. You know, if you have IBV outbreaks, if you have ILT or MG outbreaks, that will aggravate coryza, and sometimes that will show as coryza, even though you have other underlying problems. And also additional work needs to be done to understand available vaccines. And the recommendations are: review your vaccination programs for respiratory diseases, check on your IBV, ILT vaccination program, make sure that those are adequate to the challenge that you have. How do you know the challenge? Doing surveillance for these diseases, specifically for IBV, that's extremely important. Audit vaccination crews and check for vaccine takes. Vaccinate against coryza with 2 doses of killed vaccine, looking for shedding reduction and environmental challenge reduction. And that's, I want to use maybe 30 seconds to explain why 2 doses are important. When you apply only 1 dose, the immune system is not able to generate good memory. These vaccines are killed. So what they are stimulating is the production of antibodies, right? When you apply it once, that amount of antibodies is not enough in order to prevent fully the clinical signs and also prevent shedding in a good way. It will reduce the amount of bacteria that is shed in the environment, but not at the point in which we need in order to reduce the challenge, the complete challenge in the environment. So the only way in which these vaccines will work, masking or eliminating clinical signs and reducing the challenge in the field, which is the end goal, right? I mean, we want coryza to be less in the environment so we don't contaminate other flocks. We want to be a good neighbor. For that, you need to apply the vaccine twice, disregarding the vaccine that you're using. And with that, I would like to acknowledge my lab and funding for this experiment. And here's my contact information, my email. Please contact me if you guys have any comments, questions, or if you guys want us to go and check your vaccination crew, or you want to discuss about your vaccination program or your vaccines in regards to Coryza. Thank you.